This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 209, recorded on October 31st, 2019. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Joining me today, which happens to be Halloween in the U.S.
Are you properly disguised?
I'm dressed as a scientist.
That's a nice disguise.
From Small Things Considered, Elio Schaechter. Welcome back. We missed you last time. We missed you in Georgia.
Well, I'm glad to hear that. I'm glad you had a good time.
Actually, they told us when they first told the students that they were going to bring TWiM, the first thing they said is Elio coming. See, they want you. They want you. We want Elio. We want Elio.
Oh, God.
Also joining us
Lock
from
him up
from Ann Arbor, Michigan, Michele Swanson.
Hello. I think some of us have been hitting the Halloween candy early.
And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Yes, the Halloween candy. Well, I took the subway this morning, and a lot of people are in costume here in New York City.
Is that why you took the subway?
No, my car has got a flat, so I had to leave it here in the garage, and I'm trying to figure out how to fix it. Because I don't.
Do you have at least a spare?
I don't, because I have run flats. It's not. Yeah. I'm not supposed to need a spare, but the sidewall was. I hit a pothole and tore the sidewall, so I can't really drive too far. So I'm just sorting it out. But for now, let's talk about microbiology.
All right.
And later in this episode, we will announce the winner of the book Spore.
Ooh.
We had some entries in that contest, and we'll give it away.
But it's appropriate you're giving it away on Halloween. Spore.
Yeah, that's a good thing to say, yeah. Now, first up, Michael has a very interesting snippet for us.
Yeah, the paper is entitled Persister Cells Resuscitate via Ribosome Modification by 23S Ribosomal RNA Pseudouridine Synthase, RLUD. And the paper is by Song and Wood from Pennsylvania State University. So
Michael, when's the last time we saw a paper with two authors?
It's a long time ago.
Wow.
It's a long time ago. But this is a really cool paper. And the first three lines of the abstract really sets our stage for our snippet. So upon a wide range of stress conditions and examples.
You're quoting, right? You're quoting.
Yeah, I'm quoting. A wide range, well, I'm going to paraphrase a little. Wide range of stress conditions, for example, nutrient deprivation, antibiotic exposure, oxidative stress. A subpopulation of bacterial cells known as persisters survive by halting metabolism. The cells then resurrect rapidly to reconstitute infections once that stressor is removed and nutrients are present. However, how these dormant cells resuscitate is not understood well, but involves this reactivation of ribosomes. So why should we, the general consuming public that are listening to TWiM, or more importantly, the scientists out there, why are persisters important? Well, if you eat ready-to-use greens, whether it be lettuce, spinach, mixed greens, or whatever you have, the simple oxidative stress of running our produce through a dilute 50 part per million, 100 part per million of sodium hypochlorite can send a population of microbes resident on that produce. It's going to kill most of them, but a fraction of that population will go into this persistent state, which we know now as viable but non-culturable, or VBNC. So in today's work, Song and Wood investigate whether or not they can take the cells out of a state where all metabolism is halted to a state where they are alive. Now, I picked this paper because this is a big issue for the ready-to-eat produce market because we're getting all of these recalls. But they don't touch on this in the paper. But as I was reading this, it really came home to me as being of significance. So here's the gist of their paper. They screened a library of 10,000 compounds, asking which one might stimulate the resuscitation of E. coli out of its persistent state. What they found is their needle in the proverbial haystack. One compound, 4-bromophenyl, 2-oxyethyl, 3-ethyl tetrahydral benzothienopyrimidine, or they abbreviate this, and I eliminated all the numbers because I didn't expect the audience to be writing them down, but they abbreviate this as B-POET. And this compound could switch hibernating ribosomes back to their on state via a 23S ribosomal RNA pseudouridine synthase, which is the RLUD gene product, that in turn immediately increases the activity of the slumbering ribosomes. And this novel compound, B-POET, helped establish a link between how small RNAs facilitate the switch between persistence/no metabolism, or the VB&C state, and active growth. Now, as Elio pointed out to me when I pitched the paper to the group, the concept of persister cells is not a new one. It was first described to a subpopulation of bacteria becoming dormant due to stress, and this observation was made in the early 1940s, before we knew about DNA and RNA and proteins and what controlled everything. What we know today, now almost 80 years later, the persistence phenotype is not due to a genetic change per se. The evidence for this is that upon regrowth, persisters behave the same as the predecessors that they were, the originals. Secondly, we know that cells will eventually all undergo the stress of starvation at some point in their lives, and that persisters are unique in that they are tolerant to antimicrobials. And this is important to medicine because many of these persistent infections that fail to grow out a microbe are really driving infectious disease docs crazy because they bleed someone, they look for an infection, they don't see it. And it's because the cells are in this persistent state. And once the stressors are removed, they turn back on and they're responsible for a new infection. And
But Michael, you don't mean to imply that the persisters cause symptoms of disease, do you?
Mm-mm.
Okay.
They come back and then they cause the symptoms of disease once they begin to regrow.
Right.
Once they regrow. And the authors point out to us that there's a new reference out from 2019 by Renaud and Helene that now raise the level of persistence in a population from what we mentioned in a previous TWiM of 0.3% up to 1%. Now, so that's as background for us. Now, here is where they begin to form their hypothesis in their story. Tolerant cells utilize alternative sigma factors like RPOS, and many of us recall the cascading sigma factor hypothesis that was advanced by, I believe, Losick and company about sporulations. Remember, the sigma factor is the component of the RNA polymerase that confers promoter specificity, and you have sigma factors coming in conferring to the transcription complex which promoter to recognize. And these alternative sigma factors in E. coli direct genetic expression as their defense mechanism against things like stress, whereas persisters simply cease responding and become rock dormant. They're refractory to antibiotics, they're refractory to everything you can throw at it. And when the stress is removed, they simply come back on. And so the authors are really trying to understand what that trigger is that allows them to quickly turn themselves back on. The authors review the prevailing mechanisms of accounting for persistence from the literature. Since this is only a snippet, I'll leave that to the reader to go out and learn about the systems. But at the end of the day, it's a stressor that sends a fraction of the population into a persistent state. And the question that they are trying to decipher is, how does the cell know it's safe to resume metabolism? And the mechanism of how is largely still a theory. So here they set out to figure out simply if they could find a molecule to resuscitate the community. Now, what I really loved about this paper is how they did the experiments. So the first thing they did is they converted their entire community of E. coli into persister cells. And just think about how you would put an entire community into a persistent state. And the first thing they did is they treated with rifampicin. Rifampicin is an antimicrobial that stops messenger RNA synthesis. So this forces the cells to effectively cease metabolism. They then followed that up with a dose of ampicillin to kill any of the remaining non-persister cells that may have gotten through the rifampicin dosing. And the result of this was 100% of the community were now indeed persister cells. And they then went through great pains of conferring that the persister cells generated in this way behaved as they should.
I was also excited to see many other groups have used the same protocol to generate persisters in different bacterial species like Pseudomonas and Staph. So this is a great tool for the laboratory.
Yeah, because as we all know, it's really hard to synchronize bacterial growth. Bacteria are independent entities and they all grow at their own rate depending upon nutrient delivery and waste removal. And it's really hard to get synchrony. And this was one way to send the whole population into that venue. So they next then screened a library of 10,000 compounds that would enhance persister cell resuscitation. And the way they confirmed this ultimately is via single cell microscopy. And they literally watched individual cells and they learned that persister cells wake via ribosome activation by activating chemotaxis, and showed that these VBNC cells or VBNC cells are equivalent to persister cells. So they then did something really clever, and we already told you they found their magic compound, B-Poet, to resuscitate the E. coli. But then here's where their heavy lifting comes in, and this is where Vincent's original comment of only having 2 authors comes into play. This group really did a lot of work. They pooled 4,267 Asca clones, and an Asca clone is a complete set of the E. coli K12 open reading frame archives. And if you're interested in Asca clones, there's a paper by Kitagawa that was published in 2005 that describes this whole business of expressing all of the open reading frames out of E. coli K12. And I'll put that into the show notes if you're interested in learning more about Asca clones. So Asca clones, this is where each E. coli protein is produced. And the way they did this is from a plasmid. And the rationale behind this selection scheme, if you will, was that any pathway stimulated by this magic compound, B-POET, would be even more active if the number of rate-limiting proteins in that pathway were increased, and that the cells that wake first would form colonies faster on a Petri plate. And what this screen enabled them to do
I tell you what, you want to go over that once again. It's a little bit complicated.
It is indeed. And I didn't know if I was going to get the time since it's a snippet. So they have these open reading frame clones that are producing each and every one of E. coli's proteins. The plasmids are in E. coli in these persister cell population. So you have these plasmids that are now off, and then you turn them on with the B-POET. The B-POET effectively gets the cell out of its persistent state, and it now will turn on. But before you turn them off, the plasmid is in there overproducing all of the proteins of E. coli. So the 4,267 Asca clones are each one of the open reading frames of E. coli. So they're overproducing the proteins ahead of time. You then put the population in the persistent state, and then you ask how B-POET could unlock the persistence trait and turn it back on. So their hypothesis was that the cells that wake first, that have the extra gene that is involved in persistence, once B-POET turns on the systems again, those cells would get bigger faster, so they could effectively pull it out of their screen. It's simply when you look at your Petri plates.
But they were actually looking for a pathway that would stimulate resuscitation, correct?
Correct.
Mm-hmm. Right.
And that is effectively how they selected the process of what gets the cells that wake first would form colonies faster.
So they're potential targets for their B-POET.
Correct. So you already had the targets in there. You had an extra copy from your plasmid, and it was probably the garden variety multi-copy plasmid, so you have many more copies of the 5 identified proteins which they found. And the one that they characterized as being the one that was involved in the ribosomes was the RLUD gene, which is the 23S ribosomal RNA pseudouridine synthase gene. And another one was the SRLR gene, which represses the gut operon for glucitol metabolism. And they characterized them, and they focused on the RLUD gene because it related to ribosomes. And part of their background information that I didn't share with you is that their fundamental hypothesis is that ribosomes were the gatekeeper for what gets a cell out of its persistent state. They next then take the reader and help you appreciate what the RLUD gene product, or this pseudouridine synthase, does. And it's involved in the synthesis and assembly of the 70S ribosomes, as well as their function based on the post-transcriptional modification of a 23S ribosomal RNA product to form 3 pseudouridine nucleosides at 3 particular positions in that 23S ribosomal RNA molecule. which meant nothing to me at the time since protein biochemistry was a long time ago. But the authors graciously helped me remember my protein translation in that pseudouridine uracil. The uracil is attached via carbon-carbon bonds to the sugar base rather than a carbon-nitrogen bond. And the 23S ribosomal RNA molecules, pseudouridines, increase the stability of the tertiary structure of 23S ribosomal RNA, which are located in the stem loop structure that is involved in the peptidyl transferase, moving the amino acid into the peptide, which then serves to interact with the messenger RNA, the tRNA, the 16S RNA, and the ribosomal release factor, which is simply code, is that RLUD is responsible for efficient ribosome function. So you have to remember some of your protein biochemistry and what this fancy gene product is actually doing, getting the cell out of the persistent state. The next bit of their paper was just plain elegant. Here they use single cell studies as a consequence that the synchrony in populations of bacteria is just plain hard. And so they watch one cell at a time. And they first then deleted the RLUD gene from a strain, and this greatly reduced the ability of persister cells to recover, and enabling them to ultimately conclude that this gene, RLUD, enhances persister cell resuscitation. Then, in a clever experiment before making the community of persisters with their RIF amp trick, they treated the community with Be Poet ahead of time before they did the RIF amp trick. And they learned that over 85% of the cells had a higher ribosome fraction compared with cells that did not overproduce the RLUD protein. And, you know, it was just phenomenal. They just walk us through how this molecule enhances RLUD production. And they then talked about how another gene product antagonizes persister cell resuscitation. And that particular gene product makes a small RNA that then in turn reduces the RLUD gene product. And as you might expect, it decreases the waking function and it by at least 8-fold. So putting this all into perspective, could we simply add Be Poet to the contents of the material that we test in produce to determine if produce is safe, to help us figure out if there's any viable but non-culturable cells in our ready-to-eat produce, really revolutionizing food safety. So this paper is a fundamental piece of mechanistic science. At the same time, you can begin to see how it could have practical applications in the long term, addressing the whole food safety business, as we're beginning to appreciate that VB and C cells and persister cells are important in releasing produce is safe when indeed it does have viable bacteria in it. Recall that we did a paper, one of the earlier TWiMs, specifically about persister cells and bleach and other things, and that was in Listeria and Salmonella. That was earlier this year.
Let me ask you, since persistence is not just a produce problem, but it can be a problem of people as well in certain infections, is Be Poet safe to use on people? Anybody?
That I don't know.
Hmm.
That I don't know, but I'm that's why I picked the paper, because it's sufficiently provocative enough. The authors in their figures show us the chemical structures of these molecules, and not being a medicinal chemist, I don't want to comment one way or the other whether or not you might be able to give the bromophenyl oxyethyl, B-poet molecule to it. But you bring out an important point, because in prosthetic joint infections, oftentimes you can't culture a bacterium out of that infected joint. But if you do next-gen sequencing on it, you can identify a whole slew of microbes that are present in there in a significant number, yet you can't grow them. And yet there's significant pathology in the patient because your immune system is recognizing the presence of the microbes, and they're trying to clear them out. So you put a really good question on whether or not B-poet could be used to effectively turn things on so antibiotics would knock them out.
Another potential application is in TB, where long-term persistence of non-replicating or slowly replicating bacteria are hard to clear by antibiotics, and you get reactivation disease. So it'd be interesting to know if it could be used there. Or I'm thinking in water treatment protocols for getting rid of Legionella.
Yes.
That is, hospitals will have a problem with Legionnaire's outbreaks. They'll remediate, and then months later, they'll get regrowth, thought to be due in part to persisters.
Yeah.
Right.
Yeah, it's a good idea. Michael, is there any reason to think that this particular mechanism involving the ribosome would account for persistent in other than E. coli?
Oh, no. I think the protein biochemistry is pretty much cut and dried across the bacterial world, because the broad spectrum anti-ribosomal drugs work on a large number of different bacterial species in general. Mm-hmm. So I think, this is a fundamental mechanism that I think the cells have evolved over time to, if you will, keep a genetic presence in times of hardship so that the population can ultimately rebound when you produce nutrients for them or you create a more favorable environment as the stressors are removed.
All right, so that's a yes, right?
Yeah, that's a yes.
Although we don't know what the direct binding target is of the B-poet. So it remains to be investigated.
Yeah.
But this was only 2 people, and they did a lot of work. And if they had 30, maybe more.
Yeah, I think there's more work to be done here, but pretty interesting.
Yes.
Really neat. All right. Thank you, Michael. All right. Now we have a paper from Elio.
Well, I'm changing gears, but good. We're talking about phages. Now, in this podcast, we usually stay away from viruses because our glorious leader, Vincent, runs his own virus podcast, This Week in Virology. But somehow he's allowed us to talk about viruses in This Week in Microbiology. Very nice of you.
Well, you can always talk about phages here. It's no problem.
Good. So let me wax lyrical first.
Are you going to wax poetic, Elio?
Poetic too, and lyrical.
Be poetic.
Here we go.
Oh.
The world of phages is strikingly diverse. Some phages use RNA to encode the genome, others DNA. Some are tiny, others are 1,000 times bigger than that. Some are bare icosahedrons, others are decorated with all kinds of fancy threads. And each picks its own favorite replication strategy from a large menu. So this is some world. In addition, viruses are the most abundant biological entity. Never mind if they're alive or not. I'm not going there. They're the most abundant biological entity in that they are 10 to the 31st phage particles in the world, mainly in the sea, in soils, everywhere. They are everywhere. And they have been resuscitated lately because they are, of course, they've been known to be useful in treating disease, bacterial disease. but it's a complicated story, which we touched on that, didn't we? Seems to me.
Yes, sure.
Yeah, we
talked about phage.
Right, we talked about phage. Anyhow, never mind that. So I would suggest for people who want to know more about phages that they look at a fantastic book written by my ex-collaborator, Mary Ure. It's called Thinking Like a Phage. Can I quote a sentence from her? She says, although outside our normal vision, the phage multitude is always there, an ancient, dynamic, bubbling, creative force that underpins all life on Earth. How's that?
Wow, that's a bold statement.
I've done enough waxing, okay? All right, let me now tell you that if I had to choose one kind of phage that is my favorite, I would choose a kind that's called flagellotrophic. That is, they go towards flagella. They stick to the host flagella for the initial step in their multiplication. Now, I think this is one of the most surprising things that can happen because all phages, all viruses, stick to a receptor on the cell envelope, the cell membrane in the case of eukaryotic cells, the cell envelope of complicated cell envelope of both gram-positives, negative bacteria. But these guys stick instead to the flagella. Now, how can that be, and why is this so exciting? Well, first of all, it's not
whose beautiful work are you describing? I'm not sure we've heard the title yet.
We've just heard waxing.
Thank you. I got so excited.
I know.
Thank you so much. The title it's from an article in Molecular Microbiology on the flagellotrophic bacteriophage YSD1 targets Salmonella typhi with a chi-like protein tail fiber. And the authors are quite a few, unlike the other paper. Dunstan, Picard, Dogan, Goulding, Corney, Hardy, Grinter, Harcourt, Song, Schreiber, Chaudhary, Clare, Kulbali, Strugnet, Duncan and Lithgow.
And also Lee, Yu, Chaudhary.
Did I mention it?
Chaudhary. Yep.
Okay. And they are all from Australia except for two people who are from Cambridge, England. Thank you. I'm sorry about having left that out. Okay, so the paper, I'll come to the paper at the end, but I have to really talk a little bit about flagellotropy in general. So in the title they say this is a chi-like protein tail fiber. Chi here, which is spelled C-H-I or the Greek letter chi, depending on what area you're in, is a phage that was discovered in 1936. So it's an old story, and it was known, and this is from the laboratory of Harel, the guy who, together with Dwart, discovered bacteriophages. This is in Paris. They soon found that it only infects host cells which are both flagellated, they have flagella, and they're motile. If there's no flagella motility, there's no infection. This turned out to be very handy because if you look at chi-resistant mutants of Salmonella or E. coli, you can isolate mutants which are defective in flagella formation or flagella function. So the study of flagella benefited from looking at chi-resistant mutant. We clear about that so far? Okay. When the electron microscope became available in the in the early 40s, really, they could tell that chi-phage particles attach to the flagella, and they attach by a long single fiber on their tail. It's a fiber that's sort of turned around, kind of kinky, okay? Now, the people who discovered this, or soon afterwards, some people who studied it thought, hey, maybe the phages get into the flagella and use it as a tube. Turns out not to be the case. That's a good idea, but it wasn't true. What happened is they slide down the flagella filament towards the site of infection. And they reach the bacterial surface where there is a classical kind of cell envelope receptor. And now they can deliver the DNA and they start the multiplication. Now, we had this written up in our blog, Small Things Considered, where Michael Yarmolinsky once wrote, and I'd like to quote this. He said, as a macroscopic observer, I view the ability of Chi to lasso and ride astride a spinning filament as an acrobatic feat out of the Wild West.
It's definitely a rodeo. It's definitely
the details of this rodeo could be interesting. That's what Yarmolinsky said. So anyhow, you agree with me? This is interesting, right?
Yes,
absolutely.
Certainly.
So they found flagellotrophic phages are not just found in Salmonella and coli, but also in bugs like Aeromonas, Campylobacter, Pseudomonas, Colobacter, and Bacillus subtilis. And in each case, the interaction is specific. It takes a kinship between bacteria and their phages. The phages are specific. Well, that's obviously. Anyhow, most phages that do this, that are flagellotropic, have one fiber sticking out of the tail. Others have three. But one, a Colobacter phage, beats the band. It sticks not by the tail fiber, but by a single filament that emanates from the capsid, from the head, because they attach head first. Now, strange, and it's really strange. This is a strange world of flagellotropy. Now, let me make an aside. Flagella are not the only thin, long appendages of bacteria to which phages can attach. Pili count also. There are some bacteriophages that stick to pili. So why is that? So people think that by using the filaments that are sticking out of the bacteria, the capture radius, they call it, that is the effective volume on which phages can be found to attach to bacteria, increases tremendously. So this is a way of increasing the frequency of successful encounters between phages and bacteria.
Because the phage themselves are not motile. They're just drifting about.
Phages themselves are not motile, so they depend on random collisions, and random collisions are increased if you send out a hook, as it were. Okay? All makes sense. So why are active flagella needed? Why isn't a dead flagellum good enough? Well, first of all, there's a nice teleological reason. A beating flagellum means the bacteria are alive and well. Phages like that because they don't like to stick to a dead cell. It doesn't do any good. They will not reproduce by sticking to a dead cell. So the beating of the flagella is a signal that the bacteria are physiologically active and likely to support phage development. Pretty good, isn't it?
Mm-hmm. Makes sense.
Like
Makes sense.
Makes sense. So now that we know why they do it, how they do it, well, we know they don't go through the inside of the flagellum, so they go through the outside of the flagellum. So what people found quite a while ago, especially in Berg's lab at Harvard, but also in Julius Adler's lab at Wisconsin, is that that the tail fibers of these flagellotropic phages fit the grooves formed by the helical monomers of the flagella protein called flagellin. In other words, there's a helical shape to the flagella, and the tail fibers fit into that. So it's a little bit like a knot following the thread of a bolt.
Mm-hmm.
You with me? Mm-hmm. They start out in one place and they just thread themselves all the way until they get to the base. And now they are where they want to be. So this is kind of fun, isn't it? So what happens?
That was very useful. That nut and bolt image was very helpful for me.
That's right. It's really nice. And by the way, I didn't tell you, did I say that the book, Thinking Like a Phage, in there's a nice treatment of this whole thing with a nice drawing. So anyhow. So what happens? So now the bacteria, the phages thread themselves down. the flagella, and the flagella beat in the right direction so that the phages reach the base of the flagellum. So what happens when they reach that is, I say, they have a receptor, they inject, if you wish, the DNA, and off they go. So now replication of phages is possible. So that's the basic story of flagellotrophic phages. Now, the paper that I mentioned talks about a chi-like protein tail fiber. So what they did in the study is to isolate a salmonella phage called YSD1, and they go to a great length to show that it's really related to the chi phages of E. coli that I mentioned. So knowing that, they have a lot of basis for study, and what they do is they study essentially all the proteins of the phage. The phage encodes for 71 proteins, and 24 of them are found in the mature virions. What the rest do, they don't really know, but they're probably involved in aspects of replication that do not necessarily get packaged into the final product. This is probably it's quite typical of other phages, isn't it? So it's what all viruses do, something like this. They have proteins which are required for replication, aspects of replication, but are not included in the final virion. Am I right, Vincent? You say something?
Mm-hmm. Yes.
Okay. So now they focus on three of the viral proteins, the proteins which are included in the virion, and two of them make up the tail tube, YSD1-22 and -25. 22 makes the main shaft, and 25 is part of the tail spike. The tail fiber itself consists of protein YSD129. And there's a detailed study that led to the annotation of essentially all the proteins of the phage and vague hints as to local function. So now that the process of flagellotropy is being studied on a molecular level, we can assume a better understanding of this phenomenon. And that's about all I would like to say about it, because the paper is very detailed, but it deals mainly with all the elucidation of the properties of this phage.
I was very impressed with how they went from DNA sequence and then phylogeny to look at evolutionary relatedness. And then they went back to some simple biochemistry, running protein gels. And then they did immunoglobulin labeling to show where each of these tail proteins actually bound on the whole phage. I mean, they just went really step by step with really elegant mechanisms, and then took us right down into the protein structure and then tied it back to function.
I think the one thing that we should point out is that they did a structure of this 29 protein, number 29, which forms the Flexus Filament at the end of the tail, and it looks helical. And they suggest that the way this helps the phage slide down the flagellar cable is that it's like a corkscrew. And as the flagellum is beating, right, in a circular motion, it causes the phage to which this tail protein is attached to wrap around and around and around and move closer and closer. So it's not quite the screw and bolt analogy that you mentioned, because there is no hollow bolt to go down, but rather... Well,
nobody meant that to be a hollow bolt. I mean, it's a sort of a general...
Well, a bolt is a very specific thing. It's a hollow thing that screws onto a shaft. So this is more precise because you can see from the structure that it's a helix, and it's more likely to work like a corkscrew or go around and around and move closer and closer as the flagella beats. And that would explain why you need a living cell, because if it's not beating, it won't move closer.
Exactly.
Yeah. Not only that, but in their discussion, they point out that salmonella can either move by tumbling or they can move in a directed fashion toward food. And which they're doing depends on whether their flagella are rotating clockwise or counterclockwise. And it turns out that the interaction between the phage and the flagella, when the bacterium is swimming toward food, would be driving the bolt or driving the phage toward the cell, the bacterial cell, which is also just really beautiful. It's not random. They're only heading toward the cell body when there's going to be food, and the bacteria will replicate.
It's natural selection at work.
Yeah, and they say only a counterclockwise rotation would promote movement of the phage along the right-handed groove in the flagellum toward the cell. So that number 29 protein would fit right into that groove and slide right down it as it's rotating. It's really cool.
It is cool because it really shows a mechanism which is complex and yet simple at the same time.
I think out of deference to Vincent, we also need to mention that there are some plaque assays shown in the paper.
Really?
Where
yeah, that's good. It makes it a wonderful paper. Yeah,
it makes it a virology
use genetic
paper.
Arguments to identify the particular amino acids that correlate with binding or not binding, and being sensitive to the phage or resistant to the phage. So it's really beautiful use of that elegant assay to get right into the molecular detail.
So you contacted one of the authors, did you?
Yeah, this is actually a collaboration between labs at Monash University in Australia and the University of Cambridge. So this is Gordon Dugan and Trevor Lithgow, and it has two first authors, Rice Dunstan. He was a PhD student with Trevor Lithgow, who is an outer membrane biologist, and there he was studying secretion machineries. He stayed on to do postdoctoral studies in that lab, but had the opportunity to go do a 5-month project with Gordon Dugan's lab at Cambridge. And there he met the other first author on this paper, Del Pickard. His formal name is Derek, but he goes by Del. Del was able to take a direct path from high school into a research group. So he started at a research laboratory right out of high school and has had the pleasure of working with Gordon Dugan since 1983. So he is clearly an expert. And the two of them together were collaborating with a 19-year-old college student, Sam Dugan, who is actually the son of Gordon Dugan. They wanted to introduce him to the wonders of microbiology. And that's where this project started. They did a broad screen of collecting samples from the environment and then screened on using plaque assays for new phage that could infect Salmonella. And it turns out the one we study here was from the garden pond of Del.
That's cool.
So they knew from the literature that Elio so elegantly summarized for us that typically these phage will bind to the capsule of Salmonella. But they tried that and found that wasn't the receptor. So they were chatting with another colleague in the hall just informally. This is Fernando Schreiber, and she recommended that they try Flagella. And so it was her at her prompting that they screened a bunch of Flagella and discovered that it was a flagellotrophic virus. So that's how this project was born. And then, as I mentioned, it's just a really beautiful demonstration of how you can apply multiple techniques in a collaborative fashion to come up with this very specific molecular mechanism. They both said that they were fortunate to have great mentors. Rice was especially appreciative of being able to work with both Gordon and Del, and enjoyed seeing all the pieces come together in this story. Rice does have advice for younger colleagues. He says, try to work on something you're passionate about, and this should be true in all aspects of life, not just science. And also, just as important, look for mentors who are interested in developing young people in the field. Not necessarily your direct supervisor, but it could be somebody else in your department or faculty who will help promote your career. And then let's see, Del has also had the pleasure of working with a number of people who said were inspiring throughout his lab. William Turner, Gordon Dugan. Also, he mentions a number of other colleagues who've inspired him along the way. He also recommends that junior colleagues go to conferences, not only the ones that are right in your field, but also ones that are intentionally outside your field, because it can really open your mind and give you new insights to your work. So he recognizes the inspiration he got from William Schaaf on microfossils of cyanobacteria, John Allen, who emphasized that life on Earth would never have taken off if it wasn't for the presence of ready-made inorganic complexes in the ocean, and then also Bill Jacobs in the Bronx, who is developing novel vaccines and using phage in that research. So Dell also enjoys taking radios apart and trying to fix them, but never really doing that. And he really loves early evolutionary microbiology, especially sponges. And so he's been trying to convince the people at the Sanger Institute to sequence some sponges.
Radios. There's still radios around?
Oh, yes.
Where are they, Michael? This is next to your bed, I guess, right?
In my attic.
In your attic. Wow. That's funny because I used to take apart TVs when I was a kid.
Really?
Oh, my goodness.
I would find junked TVs, and I loved just taking them apart. And then we'd take the tube.
Figure out how to use the remote, let alone.
And then we would take the tube, big glass vacuum tube, and go in the woods and throw it at a rock.
Ooh, and it would explode.
Oh, what a blast it would make.
Yes.
Okay.
Don't try this at home, kids.
No.
I notice, Michele, there are two Reeses on this paper. Reese Dunstan and Reese Grinter. How about that?
Oh, you're right.
What's
You're
the
right.
likelihood that we would have not only two Dugans, but two Reeses?
I do not know.
Reese's Pieces. That's what I keep thinking about.
It's because it's Halloween, Vincent.
Maybe.
You must have raided the candy in the front office.
I haven't been to the front office in months.
Oh.
I can avoid it. I don't need to. Anyway, thank you, Michele, and thank you, Elio.
My pleasure.
For educating the world and us. I learned one thing in there that I didn't know, that the salmonella are endemic in birds.
They mentioned that a recent survey of a poultry farm in Thailand found that phage were the predominant type of phage, which makes sense because salmonella are common in poultry.
Right. All right. Thanks again. All right, let's give away Spore. Spore is an ASM Press book, all about spores, as you might guess. And we had 8 entries. I guess not a lot of interest in spores, but one of you 8 will win. We had an entry from Brian Evan, who said, I'm such a huge fan of the 5 TWi podcasts. Anthony, Gertrude, Patrick, Yarek, and Andy writes, Dear explainers of the inscrutable. Inscrutable, both literally and because to many people, cutting-edge science, and especially papers describing scientific studies, can be incomprehensible. Hello from Stillwater, Minnesota, where it's 40 and wet, but the rain has stopped. I'm a 57-year-old IT professional who has absolutely no business trying to keep up with the biological sciences, let alone studies thereof, but I never got over my boyhood wonder at everything biological.
Good for you, Andy.
Were I to be chosen to receive the copy of the bacterial spore, I would no doubt enjoy every rabbit hole it sent me off to. But after that, I think I would donate it to the library of the hospital I work for. For me, these topics are pure entertainment, and you teachers entertain me plenty. But I'd like the book to be where it can benefit real people. And the last is from Emily. Good afternoon, respectable TWiMcast. I would like to enter to win the Spore book. I work at an Army microbiology lab and would love to incorporate the information in this book into my research. So not only are we inscrutable and we are respectable. Not a bad combination.
That's good.
All right, let's pick a random number between 1 and 8. The number is 7.
Andy.
Well, Andy, you got it.
Andy. All right.
Now, Andy, just send me your address, twim@microbe.tv, and we'll ship out Spore. You can go down rabbit holes, and then you can donate it to your hospital library. Hospitals have libraries, is that right?
The better ones do.
Okay.
In Stillwater, Minnesota.
Where it's 31 degrees and cloudy.
That's right. All right, that's TWiM 209. You can find all the notes, papers, links that we talk about at microbe.tv/TWiM. And you can send us your comments and questions to twim@microbe.tv. If you really enjoy what we do here on the TWiM and all the other podcasts of this Microbe TV family, can try and support us financially, go to microbe.tv/contribute. And there you could give us $1 a month via PayPal or Patreon. We sincerely appreciate that. Today's TWiM has included Michele Swanson from the University of Michigan. Thank you, Michele.
Thank you.
From Small Things Considered, Elio Schaechter. Thank you, Elio.
Thank you. It was a pleasure.
From the Medical University of South Carolina, Michael Schmidt. Thank you, Michael.
Thanks, everyone.
And by the way, the Our Georgia Tech episode has been posted today.
Ooh.
On Halloween, and you can see the video.
We have a video, too.
Wow.
Oh, boy.
And everyone looks lovely on that video.
Can I also congratulate you, Vincent, on the launch of This Week in Neurobiology?
Oh, thank you. Thank you very
Wow.
much.
TWiM.
Another one?
Another
Another TWiVo or
Another Twix podcast. That's
Twix.
right.
It's an X.
That's
Twix,
great.
Twix. Thank
Are you
Michele.
actually on that? Or I saw you named 3 other people, but are you part of the team?
Well, so I was approached by a neuroscientist here, and he wanted to do this, and he did all he recruited the team. He's doing
Right.
And I said, I could just record and not do anything. He said, no, I'd like you to ask questions because you would be the outsider so I'm participating.
Right. Keep them honest. Great.
Yeah, so that should be fun. I'm going to learn a lot.
You're a hero.
And I have one more idea left for a podcast, and that's it. That would be number 7, and I will wait a while to start that. But
one a day.
Yeah,
one a day.
I know they're not all. Most of them are monthly, as you know. So it's not that bad.
Is it a secret?
What's that? The new one?
The topic? Yeah.
It would be about infectious diseases in general, not just one kind, but all kinds.
I see.
And more of a historical. So I'm thinking of doing it would be episodic, where there would be a series of half-hour stories about one infection from the beginning to where we are now.
Right.
That'd be cool.
Like polio or tuberculosis or trichinella, just one at a time. You run them, and then that's a season. It's like a TV show. You would do a season on polio, right?
A season on polio.
So that's a little different from what we do now, but I would like to try it because I think it would be fun.
Well, I know the first we can get started on that with my colleague Janet Gilsdorf, who's just published a book on the history of Meningococcus, I believe.
Ah, right. We have to get Janet on TWiM to talk with us about that. Yes, we'll arrange that. Yep.
Great.
I want to thank ASM for their support of TWiM and Ronald Jenkins for his music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.